All right, so let's go ahead and go over it. Hands. Who has an answer to what the three properties of DNA are? All right, so no one? Yes.
Holds all the genetic info.
Very good. So your DNA is going to hold all of the genetic information for every cell in your body. So it holds all genetic information. So you can consider it a library. You can consider it a cookbook. And on a little side tangent, right? What is the whole point of this DNA? What is it helping us do? Very good. Very good. It's helping us make proteins. Cool. All right. So that's the first property. The second property is it's able to replicate. All right. That's right. And the importance of replication is what? Why do we need to be able to replicate our DNA? We need to be able to replicate our DNA so we can pass it on to future generations. Three.
Able to undergo rare mutations.
Yeah, so able to undergo mutations. So what big process is basically a result of being able to undergo mutations? Mutations allow us to do what? It's a pretty important part of life developing over a long-term span. Evolve, right? So we're able to mutate DNA, and because of that, we're able to evolve. Okay, cool. Let's keep on going. So the next question is, what is the backbone of DNA made of? Can anybody help me out with that? Yes.
Alternating phosphate
Perfect. So, when you think about what is DNA, right? We know that DNA is a polymer. What is the monomer of DNA? What is the monomer of DNA? Yes, it's a nucleotide. So A, T, C, G, right? Those are all nucleotides that we use to build together to build a single strand and then a double strand of DNA. So that's a simple unit. So when we draw out a single nucleotide, the thing that it has is it has a sugar, right? And the name of that sugar is what? So DNA, right? So it's deoxyribose. It has a nitrogenous base, so that nitrogenous base is going to be the A, the T, the C, or the G, or the U, right? If we're talking about RNA. And then finally, it's going to have what? A phosphate. So right there. So basically what happens, right, is you have this alternating subunit where you have this phosphate and it's attaching to the bottom of a sugar molecule right here. A, T, C, G, U. Okay. So when you think about, if you take out the bases and you're not looking at, the inside, the backbone, right? So we think about our spine. Our spine is in the back, right? Right here, what is that made out of? It's going to be made out of this deoxyribose and our phosphate. So the alternating subunits that make up the backbone of DNA are going to be a phosphate and then a deoxyribose. So it goes deoxyribose phosphate, deoxyribose phosphate. So that's what's going on there. Backbone. Cool, and you can see a picture of it right there. Now let's see. What is the monomer of DNA or RNA? We just talked about this. Yes? That's perfect, yeah. So when you think about it, right, what is the big macromolecule class of nucleotides? What do you call the whole umbrella, the top of it? You'd refer to it as nucleic acid, right? So right here you start out with the macromolecule. And then you go to the polymer, and then you go to the monomer. So, the macromolecule is going to be nucleic acid. The polymer is going to be DNA or RNA. And then the monomer for both DNA and RNA is going to be a nucleotide. Perfect. Cool. So, for the question, what is the monomer of DNA or RNA? It's going to be nucleotide. And what is a nucleotide? When you look right here, it's going to be just one subunit of one of these. So it's going to be a phosphate, a deoxyribose, and then your nitrogenous base, the A, T, C, or the G. Okay? So I'm going to go ahead and label that real quick. Nitrogenous base. Cool. All right, so what are the four nucleotides of DNA? What pairs with what? Okay, so let's talk about that. Switch pages. Also, I'm going to post all of these online, okay? So you will have PDFs that are available on my website. So if you're worried about writing everything down, don't worry about that. Worry about understanding the material. That's what I'm more concerned about. I'm going to post these same exact notes, so you'll have access to them. All right, what are the four nucleotides of DNA? What pairs with what? Who can tell me what the four nucleotides of DNA are? Yes?
Iodine, cytosine, guanine, thymine, and then A goes to G, and then C goes to G.
That's right. So A is going to go to T, T is going to go to A, C is going to go to G, G is going to go to C. Perfect. And this is for DNA. And then for RNA, what we're going to see is that A is going to pair with U, U is going to pair with A, C is still pairing with G, G is still pairing with C. Okay, really quick, why did I put two lines between the A and the T combo, same with A and U, and then three lines between the C and the G? What holds our nucleotides together? What bond? Yes. Hydrogen bonds, very good. And we're going to talk about that. So let that be in the air, right? So when you look at A and T have two hydrogen bonds between them. And that's what holds them together. When you think about G and C that has three hydrogen bonds together, that holds them together. So that's what's going on right there. Perfect. Cool. All right. Our next question is RNA, what pairs with what? We just talked about that. Base pairs are connected by hydrogen bonds along the axis of the DNA molecule. So I want to go ahead and show you this picture, right? So right here, and you're only concerned with the top right now. Let me go ahead and cover up the bottom. So right here, these are nucleotides, right? So you can see right here, here's the phosphate, right? So here's your phosphate. This right here is your deoxyribose. Is everything okay?
Yeah.
Okay. And then right here you have your nitrogenous base.
What'd you do? Who was that?
Okay, cool. So you can see right here the anatomy continues on for a nucleotide. You have your deoxyribose, which is your sugar. You have your phosphate, and then you have your nitrogenous base. Now on the other side of DNA, because we know that DNA is double-stranded, you have the same thing going on, right? You have your phosphate, you have your deoxyribose, and you also have your nitrogenous base. Now, I asked you, or I asked earlier, right? We got a good answer. What is the thing that holds nucleotides together? It's those hydrogen bonds. Okay, so what I want you to think about right now is water. When we drew out a water molecule, we had H2O, right? In our H2O molecule, what had the positive charge and what had the negative charge? The oxygen had the, yes?
Negative charge.
That's right. And the hydrogen had the,
Positive charge.
that's right. Okay, cool. So the reason why water tends to stick together is because when you have a whole bunch of water molecules next to each other, Thank you. Do I need to? Yeah. Really? Okay. All right. So when you have these waters right here, they also have this charge. So what you get is you get an association between the negative and the positive charges right here. Remember, opposite charges are going to be attracted, and alike charges, the same charges, are going to be repelling. So when you look right here, positive is going to be attracted to that hydrogen. So when you look right here, look, you've got an oxygen coming off this thymine, and you have a hydrogen coming off of this adenine. That's crazy. What I'm telling you right now is that oxygen is going to have that negative charge, and hydrogen is going to have that positive charge. So when you think right there, the same trend continues. When we look at water, we see a hydrogen bond between the oxygen and the hydrogen right there. When we look right here at thymine and adenine, you see a hydrogen bond between the oxygen and the hydrogen. Right here, when you look at the periodic table, you can take note that oxygen is number 8 on the periodic table, and that nitrogen is number 7. Number 7. So they're right next to each other, meaning that they have similar properties. So for all intensive purposes, you can assume that nitrogen is just like oxygen. So it is also going to have a negative charge. Hydrogen is right here. You've established 2 hydrogen bonds. Right here, when you look at cytosine and guanine, you'll see that hydrogen has a positive charge, right? Negative charge. We have our negative charge, positive charge. We have our negative charge attracted to the positive charge. You have learned this semester why DNA sticks together at an atomic level due to charges. And you know that because you can determine what is going to be positive, what is going to be negative, and you know that those things are going to be attracted. That is very powerful information, okay? So that's really cool stuff. Let's go ahead and move on to the next one. Let's see. I want you guys to go ahead and draw out a picture of this, okay? Okay, I saw faces drop. Let's see. So go ahead and try to tell me or try to figure out, maybe draw a really quick picture. Try to tell me where the DNA is, the RNA polymerase is, and I'll make a little word bank, and then I'll fill out your picture. Thank you for waiting. So you're going to tell me where the DNA is, the tRNA is, the mRNA, the rRNA, the codons, the anticodons, the amino acids, the RNA polymerase, and the nucleus. Cool. Is that everything? Let's see. to go ahead and give you guys probably about 3 minutes. You can talk to your partners if you're at home watching this. Try to pause this, right, and label this picture using this word bank right here. Cool. All right, go ahead and get started.
I just need this to sit down. Ellie's looking right there. Oh, you give it back? Thanks, Ellie. Oh, yeah, it's right here.
I'm being an artist right now. That's a fact.
What?
I'm being an artist right now. It's a fact.
Sounds good.
I love seeing.
Wait, is this going to all the classes? So I can like say hi to my friends?
I want to give a shout out to you.
Who should I give a shout out to?
Jordan drilling from the paper.
Shout out to my boy Jordan and McKay.
Thank you.
Isn't Ava in a class?
Oh yeah, Ava and Allie.
Shout out Ava and Allie. Yeah, that's it.
Oh, I want to be in that class.
Just say, can I go to
Are they all in the same
I
class?
love both of them.
Yes,
Hey, he said yes, go.
I forgot about that.
I'll send you a.
Wait,
It's already. It's not gonna fit on the whole.
12 days till Christmas. Birthday's irrelevant.
Birthday's not relevant.
I'm at 1. I'm at 0. Cool. All right, sweet. So we're going to go ahead and cut this short, and I'm going to help you guys label this. So this is what we're going to do. This top one right here, what is happening? What is this structure called? Yes? It's called DNA. That's right. So we have DNA right here, and then DNA is being split and replicated into what? Now, what is the thing that's making a copy of that DNA and splitting it? Yes?
never mind.
Okay, side question, so we're not going to answer that one right now. What is this thing that's coming out that's going to meet later on with the ribosome? Yes? That's right, it's mRNA. Cool. Now I want you to think, mRNA is a kind of RNA, and to polymerize something means to make something, right? So when you think, what is making RNA? What is polymerizing RNA? What is the vocab term that would come to mind? Yes? That's right, RNA polymerase. So that's where it all comes down to. So in this process called protein synthesis, we're making proteins. Well, the first step is making that mRNA inside the nucleus, right? So the thing that's making that RNA is literally called RNA polymerase. So this guy right here, right, is going to be called RNA polymerase. And that's this little circle right here, and that is making mRNA. Thank you. All right, mRNA is right here. What is the name of this first step of protein synthesis? There's 2 steps. This is the first one. This is making a copy of mRNA. What do we call that?
Translation?
It's the other one.
Transcription?
Transcription. Very good. So this whole process of making mRNA is called transcription. Okay, cool. All right, so let's keep on going on. Cool. All right, so our mRNA is going to leave, right? We've got mRNA continuing down here. Now, our mRNA is going to meet up with a couple things, right? What is it meeting up with? So, what are these things coming in? What do we call this structure right here? And I'll take a hand. Yes? tRNA, that's right. This whole structure is called tRNA. What do we call the top thing? Yes.
Isn't it anticodon?
No. Yes. Boy in the back.
Amino acid.
Amino acid. What is the thing down here? Yes. In the pink.
Anticodon?
That's right. Cool. Alright, so right here, this entire structure is called a tRNA. This thing on the top, remember tRNA, the whole purpose is to carry these amino acids. Right here at the bottom, the way tRNA knows what to match up to, right? If you're carrying something, you got to know who you're bringing it to. This is how you're able to match up. These things on the bottom are called anticodons. Cool. So your anticodon is going to come on in to the situation that's going on here. So mRNA and tRNA are matching up at what site? There's a structure. What is the name of that structure that they meet up at? It's another type of RNA. Yes. Yes, and what's another name for rRNA? Ribosome. That's right. Is it still going? Okay, good. All right. Keep that up. All right. So yeah. So your anticodons are right here. Your mRNA is right here. And what they're doing is they're meeting up at this location right here, where assembly occurs, and that location is called rRNA or ribosome. Excellent. Okay, cool. So, yeah, let's walk through the narrative really quick. Also, we've labeled everything else. We've labeled DNA, we've labeled tRNA, mRNA, rRNA. We've labeled the anticodons, the amino acids, the RNA polymerase. We know that the nucleus, right, is this big old bubble right here, where everything is being held, right? So that's the nucleus. And we have yet to label protein and codon. So really quick, where are the codons located? Yes.
It's that very bottom square.
This one?
Yeah.
Yeah. But can you define what a codon is? It's a group of.
The group of.
It's a group of 3 that is on the mRNA. Yeah. So when we look at the mRNA, right? Our mRNA is this long strand. A codon is just when you split the mRNA information into 3 pieces, or into groups of 3. So, for instance, right, this right here is a codon, and that's on the mRNA, and that is matching up with the group of 3 right here on the tRNA anticodons. So what's happening is you have mRNA going inside this ribosome that's holding it all together, and you have to bring in amino acids. How in the world do we know which amino acids to bring in? Well, what happens is this coding right here on the mRNA, they're all specific and they all have codons, right? So what happens is the anticodon matches up with the codon, and then because that anticodon is matching up with the codon, it brings in the appropriate amino acid up top. All of that is held together by the ribosome right there. Okay? So that's what's going on. Thank you. All right. Sweet. Literally. All right, cool. So that's that. Yeah, that's where the codon is. And then finally, the thing that we're building right here, what is this called? This is the entire point of this process. Yes?
Proteins.
Yes, it is a protein. All right. Figure that out. Good job. That's a protein. We labeled the codons. Sweet. So I will post videos of this online. So like a YouTube video not made by me that's really good at explaining all of this. But, you know, we just walked through it. If you have more questions, ask me. Cool. All right, moving on. Our next question is, oh yeah, first off, what is the name of this whole thing that happens at the bottom? We did transcription. The next step is?
Translation.
Translation. All right. So very quickly, let's go ahead and define what the 3 steps of translation are. So we're going to keep on this topic, right? So right here, we're going to label this 3 steps of translation. steps of translation are going to be these. Let me go ahead and find my image. So right here, these are the 3 steps of translation, right? 3 steps of translation. So that's what's going on there. And this is where you have mRNA meeting up with the ribosome, meeting up with the tRNA. So the first step is where the mRNA and the ribosome and the tRNA are all matching up, and the first amino acid is added. So what you could call this first step, right? The things that are happening include rRNA and tRNA and mRNA assembly. The second thing that's going to happen, right, is you're going to add the first amino acid. Now, what do we call this first step of translation? This first step is referred to as initiation. Very good. So this first step is called initiation. Perfect. All right, so that's the first step, right? The second step is going to be right here, and in the second step what we have is the continual growth of amino acid chain. Cool. All right, so what do we call this step where you're just continuing to add amino acids to build up that chain? We're going to call that elongation. Perfect. And in this final step, we get to the last amino acid, which is going to be called termination. So in this last step, we end the growth of protein chain and the rRNA and tRNA and mRNA all separate. So they disassemble, right? Cool. That is that. Sweet. So those are the 3 steps of translation. So now we've gone over protein synthesis. Let's keep on going. Let's see. Functions. So really quickly, what do functions do in our body? What do proteins do in our body? What's the whole point of having them? Yes. That's perfect. So basically what happens, right, is proteins, what they're good at, proteins are excellent at basically doing all work in body. Almost all work, right? Perform all metabolism, all in quotations, not all of it, but basically a lot of it. Not all of it. Perform all metabolism in body. So this could include, right, acting as a catalyst. It could include, let's see, what other things would it be included? Moving substances from one part of a cell or one part of the body to another. Cool. So that is the function of proteins. Now, really quickly, what do we call the joining and the separating of amino acids? The joining of them is referred to as this kind of reaction, or this, yes?
Condensation.
Condensation, that's right. So the joining of amino acids is referred to as condensation. The separation of amino acids is referred to as hydrolysis, right? So if you're thinking, right, how do I remember that? Well, to lyse something means to break it. When you lyse something, you break it apart. And you do that with water. So you're using H2O to break stuff. Hydrolysis. Lysis meaning break, hydro meaning water. Condensation is the production of water. So if you're releasing water, right, then you're basically making a bond. You're doing the opposite of hydrolysis. So that's more of just like a memorizing kind of thing. Okay? Let's keep on going. Let's draw. So we do monomers, function of proteins. Monomer. What is the name of the chemical reaction? Okay, cool. Let's keep on trekking on. Let's take a look at this really quick. Okay, I got it. So now we should go over, right, what is a start codon and what is a start amino acid. So when you look at this chart, this should look familiar. The starting amino acid for every protein chain is methionine, right? So MET. So you should know that the thing that starts off every protein chain is a methionine. And it's kind of interesting why, but we won't get into that. Now, what would be our codon for MET? And I'll take a hint. AUG. That's right. So you can bet that every time you see AUG inside your mRNA, you're going to get MET. And you're going to be starting an amino acid chain, which is a big deal. So let's go ahead and write that down. So for every protein built, it starts with an amino acid called methionine, or MET. The codon for MET is AUG. And we know that by looking at our codon chart. The next part is, right, what would our DNA be? So if this is our mRNA, what would our DNA be? Yes? That's right. So we know that A is going to pair with T, and now we're in DNA, right? So we're allowed to use T's. So we're going to have T, and then U is going to pair with A, and G is going to pair with C. So in your DNA, a start codon will be a TAC. When you get into the mRNA, which is the thing that's being made from DNA, you're going to get AUG. This AUG is coded for methionine, and that is the start amino acid for every protein chain. Okay, let's keep on going. Let's see, so, yes, perfect. So what are the 4 full names of nucleotides in DNA? We're going to go ahead and go back up here, right? So what do we call the A, what do we call the T, the C, and the G? You know? Yes. That's right, adenine. T is going to be called, that's right, thymine. The C is going to be referred to as, yes? Cytosine, that's right. And the G is going to be called what? Yes? Guanine, that's right. And then for RNA, that U is going to be called uracil. That's correct. Okay, cool. So we've gone over that. And we know what RNA polymerase does, so that's good. So we're going to go ahead and go down the line, right? We talked about RNA polymerase. RNA polymerase is the thing that's making mRNA from DNA in the nucleus. Let's talk about introns and exons, right? So one thing that's kind of cool is when you look at DNA, what you'll find out is that there's junk DNA, and then there's DNA that actually helped us out by making proteins. So what happens, right, is when you have your mRNA that's made from DNA, what's actually inside of it are exons, and then you have some introns, and then you have more introns, right? And then you might have some more exons. So what happens, the difference between introns and exons is that introns basically do not code for DNA, right? So introns, you actually cut them out, so by the time the mRNA gets to a ribosome, all you have are exons. Introns are cut out and removed by spliceosomes. So you start with mRNA. You've just made it from DNA, from RNA polymerase. Yes? Yeah. Here, can you go out that door? And then let him back in when he comes back, okay? Alright, so DNA, you get mRNA. And this is all from RNA polymerase, right? And then you get this mRNA. Inside your mRNA, you cut the introns out. And then what you get in the end are exons. So exons are basically genetic information that's expressed, whereas introns are not expressed. So exons expressed for proteins. Introns do not express for proteins. In fact, they're cut out, right? Cool. Alright, so we went over introns and exons. So our next question is, does all DNA code for proteins? No. And why not? What is the thing that is not coding for proteins? Introns, that's right. So, not all DNA codes for proteins, i.e. introns are cut out. Cool. Now, our next question is, how does the order and number of amino acids affect the function of a protein? Think about the relationship between structure and function. All right, so we're going to go ahead and do this, right? I'll have to pause the camera for this one, so this will be nice. But what I want to do first, before we pause it, right, is I want you to consider basically things that eat bacteria, right? You remember the Pac-Mans? And we found out that Pac-Mans eat stuff, right? So our Pac-Man is going to eat stuff. Well, this right here, this whole string, the reason why I didn't lift up my pen is because this whole string started out as a polypeptide. And before it was a polypeptide, it was a chain of amino acids, right? So just a chain of joined amino acids. So what I want to show you right here is this. We're going to go ahead and pause the camera, right? So right here, just hold on, I'll do it. This Pac-Man has a specific shape, and it's made for eating ghosts, right? So we're going to go ahead and pause this. Structure, function. Okay. All right, so we're going to do this. This will be a nice break.
Let's see. How does the order and number of amino acids affect the function of a protein? Think about the relationship between function and structure. Okay, so I'm going to draw you a picture, right? So we're going to go ahead and do this. Let's imagine we have some students right here that really like each other. Oh, well, like their friends, friend group, right? So all these guys, and they're part of the basketball team, right? And we're all going to label them B, right? So there's a whole bunch of B's running around. 1, 2, 3, there's 4 B's, right? Conversely, we're going to say that there's a whole bunch of people that enjoy bicycle riding, right? So these guys enjoy bicycle riding. Right here, so we're going to call them cycling. Call them C1, C2, C3, and C4. And now, we have people that just love to talk in class, right? And we're going to call these guys the talkers, right? So, T1, T2, T3, and T4. So imagine, these are all amino acids, right? So our B's are amino acids, shh, there's no talking. Amino acids are B's, C's are amino acids, T's are amino acids, they all want to be next to each other. But if we had this long line of B's and C's, right, and T's, and then we mixed up the order randomly, so we had, another T, and then a B, and then another C, right, and then we had our third C right here, and then our fourth, and then we had T3, T3, T4, B4. Okay, so I just randomly chose right now this order. And what I want you to think about, right, is if you were inside this group, and there were 2 other groups of friends, and there was a long line, and you guys had to wait there for a while, you'd probably move around the line, right? You'd want to move closer to your friends. So what's going to happen is these B's are going to want to fold over, right? So this will probably fold over, and it's going to want to come over here. The T's are going to want to fold over, so maybe they'll come over here, so they can talk to these people. And then our B's, and then our C's, right? Our C, our 1C1 is all lonely, it's way over here. So basically what's going to happen is we have this long line, and we're comparing this to an amino acid chain, and these amino acids want to be able to talk and interact with each other. And all of that is based off of charge, okay? So, when you think about this line right here is going to be folding, and it's going to be forming a different shape. So what we might see in the end is we might see, our B1 over here, but we're still keeping the same order, right? With our C1 right here, and then now we're close to, T1, T2, and this is actually folded over on itself, so now B2 is right here. Now these guys are next to each other, but look, right? The next one after B2 is going to be our C's, so maybe our C's also came around, right? So our C2, our C3, our C4, they're just trying to be near each other. So what I'm trying to show you with all of this is you might have a long line, but when these things try to come together, basically, it's going to fold on itself. So, yeah, that's what's going on there. Cool. Let's move on. Let's go ahead and talk about the levels of structure. So, there's primary structure, there's secondary structure, there's tertiary structure, and then there's quaternary structure. So, for our primary structure, this is the level at which you just have amino acids stacked near each other. So, primary structure is going to include this right here. So it's amino acids, right? That's simple of a level. Our secondary structure is when these amino acids begin to fold onto each other. So what do you get? You have the amino acids begin to fold. So when you look right here, those are going to be like the beta sheets, right? You guys drew pictures of these. There are sheets that were forming, and also, what are they called? They're called spirals, and they're called helices. So your alpha helices. So you have your beta sheets, you have your alpha helices. And this is when amino acids begin to fold. And right up here, primary structure is just a chain of amino acids. So as you start to go from primary to secondary, you're actually like zooming in closest at primary, and then when you get to secondary, then tertiary, it's like you're zooming out, right? You get farther and farther away, so you're getting larger and larger. Now, all of these things are building on top of each other. primary, secondary. What happens next is now these sheets and these helices, they start to fold on each other, and you get a giant clump, and that's going to be called our tertiary structure. And that's just going to be like a clump, right? So that's going to be like the top part right here, right? So now you have a protein clump. Cool. Alright, so our protein is folded. The next thing that happens is going to be our quaternary structure. And, you know, tertiary, quaternary, they're very similar. Quaternary basically means that you're bringing these subunits that have been folded and you're putting them together in different pieces. So now you have these giant pieces and now you're putting them all together. So quaternary structure is like, let's say this is the top half, right? If this was quaternary structure, you'd be bringing multiple proteins together. So you'd be having a top half. And if we're concerning ourselves with Pac-Man, right? If we only have the top half, a complete quaternary structure would include the bottom half. So right here, you have your protein folded, and now you have multiple proteins. Accumulating. Cool. Alright, so our next question is, what polymer is affected by mutations? So when you guys think about mutations, right, what is the thing that's changing that is being affected, right? Yes? The thing that is going to be affected, what is the polymer that is affected by mutations? It is going to be proteins. On a more like meta level, right? What is the thing that is actually changed that results in the proteins being affected? So the thing that's actually being changed is your DNA, right? So you were right, it's just the way I framed the question. So when you have mutations, let's see, mutations change DNA. And this affects our lives because different proteins are expressed. Cool. So we got that. Let's keep going. Can mutations be passed on to offspring? Oh, is that better? Thank you. Cool. Alright, so mutations affect our lives because different proteins are expressed. Now let's say, right, our parents, they have this mutation. Can that mutation be passed on to us? Of course. So, mutations can be passed on to offspring. Alright, so the next thing that we're going to go over, right, are what are the three properties of DNA? Let's go ahead and go right here. Oh, we already went over this in the beginning. So we went over the three properties of DNA. What is an enzyme? So an enzyme is basically go ahead, yeah. That's right, so it's a protein. Okay, so we got enzyme. What is the difference between point and frame shift mutations? Okay, so now we're going to talk about mutations. Alright, so with mutations, right, you can have two types of mutations. Let's say we're talking about this giant lemon right here. So, for all you at home, we got this crazy lemon right here. This lemon right here, we're going to call it lemon gigantic. And the protein that allows lemon gigantic to be a lemon, like this, is a beta protein, right? We're going to call it beta big. And this is fictitious, this is not real. So we're going to go ahead and talk about this, right? So in your regular lives, right, you'll see a lemon. And what happens, right, is you have beta regular, right? Beta regular is a protein that allows you to get this trait, which is regular sized lemons. Okay, now when you have a mutation, you can have, and this is, typical, and this is mutation. Right here you have beta big, right? This is going to give you gigantic lemons. Cool. Alright, so when we talk about this, right, let's say that for the typical one, what we're getting is we're getting a, let's make this interesting. So, what you're getting is you're getting a proline, you're getting a, well first you gotta start out with methionine, right? So methionine, you're getting proline, and then at the end of that, you are getting a different amino acid, we are going to call it, look at the chart. you're getting lysine, okay? Now let's say this, with our mutation, what's happening in our mutation is we're getting something different, right? Instead of getting lysine, what we're getting instead is we're getting methionine, proline, and then arsenic, right? No, arginine. No, what's going on? Yeah, arsenic. So, that's what's going on there. So we have this mutation that's going on. We used to have LYS, now we're getting ASN. So that's a mutation. And these right here create different proteins. This is the protein fictitiously called beta regular, this one is beta big, and this gives us regular sized lemons, and this one gives us gigantic sized lemons. So let's say we have a DNA sequence, right? And we're trying to find out what is the one for our beta regular. So what we can see is we're going to have for our typical, our DNA is going to be this, right? It's going to be TAC, and then because we know that we need AUG to get that, this is the mRNA, right? So we have AUG, and then for proline, we look at our chart, and we see, okay, a possibility for proline is going to be CCC, right? So we're going to go and say that our mRNA is going to be CCC. And then for lysine, we look over here again, and we see, okay, for lysine, a possibility is AAA. AAA. So when we look over here at DNA, we know that we're going to have TAC matching up to the DNA, we're going to have GGG matching up to that CCC, and then over here for our AAA, we're going to have TTT. Okay, and that's what we're usually going to get. Now let's consider where our mutation is going to come into play. Okay, so right here we have our typical, right here we have a mutation. And this is the regular DNA sequence, right? So we're going to go ahead and change the regular DNA sequence a little bit. And instead of having this T at the end, instead of having a T, what we're going to have instead is we're going to have a G. Okay, so we're going to have TAC, we're going to have GGG. At the very end, we're going to have TTG. Okay, so right here, this is a point mutation, right? We're changing one of the amino acids. So we look right here and we see, okay, TAC, we look at our chart, right? Oh no, first we have to transcribe it. So we look at our chart, we get AUG, and then we get CCC, and then we get this weird thing over here, it's AAC. Okay, cool. Alright, and remember, this is our DNA up here, and this is our mRNA. Cool. So, when we look right here, for AUG, we're going to get MET, which is good. For this, we're going to get our proline, which is good. And then right here, this last one, instead of being AAA and getting us lysine, instead we're going to have AAC. So we look right here, we go AAC. Man, we were right off by one. And look, lysine used to be right here, but now we have ASN. And instead, we have this asparagine. That's what's going on right there. So, yeah. So now we're going to get ASN. And you can see, right? So we used to have MET, proline, and lysine. And now we have this ASN. So right here, we call this, first off, a point mutation, because that's what happened first. And then the second one right here, because you change the amino acid as a result, is going to be a missense, right? So right here we have a missense mutation. So when you have a point mutation, right, that's where everything starts out. There are three possibilities of what can happen. You can have a missense, where when you compare the typical versus the mutation, now you're changing one amino acid. You can have a silent mutation, where instead you're getting no change, right? So let's say we went over here, right? And our mutation, instead of getting us AAC, so AAC, let's say instead it got us AAG. Now we're still getting lysine, which is good, because that's what we used to have, but there's been a change in our DNA. So we changed the DNA sequence, we have a different codon, but despite the change, we still have the same amino acid. So what's important to know about this is you can have different codons, but still get the same amino acid. So when you have a mutation that does not change the amino acid chain, no change, it's called a silent mutation. Okay, so we have our codons different. No change in expression of protein. Okay, let's go ahead and go to the next one. So we had silent, we had missense, and now we're going to have nonsense. Who can tell me what a nonsense mutation is? What is that getting you? It's not getting you a different amino acid, it's not getting you the same one, instead it's getting you this special thing, it's getting you an early stop codon. So when you have this early stop codon, you cut that chain off at the very beginning, or somewhere early. It doesn't need to be the beginning, it just needs to be earlier than what it's usually going to be. When you cut it off early, now you don't have the rest of the chain, you've destroyed your protein. Now there's another type of variation of mutation, and that is called a insertion or deletion. Actually, okay, so we're going to take a step back, right? It's going to be called a frame shift mutation. And what's going on here, right? The cause of these are going to be insertions or deletions. So when we look back and we see that our original DNA strand was TAC, GGG, TTT, instead of having that, which you might get if we change, one of these guys right here, is you'll have TAC, GG. Remember, we're cutting off short. We're getting rid of one of the G's because it's it's an insertion. So we have our G right here, right? If we wanted to add one more in, let's say we added in right here where this T is, we can have TTG, and now we have an extra T. The deletion could be an example where instead of having the extra G put in, we have one taken out. And now we have GGT, TT. And now we're missing something. So what I want you to recognize, right, is we used to have this really nice frame. Right here we used to have TAC, GGG, TTT. But now we have this weird frame where we have, TAC, GGG, TTG. But now you have this extra T. So everything after that is going to be affected and it's going to be changed. And you're not going to have the same codons onwards. In the same way, deletions are basically going to have the same frame shift mutation. Everything after that is also going to be affected and changed. So that is what's going on there. Frame shift mutation. Okay, we're almost done. The only thing that we need to go over now is we need to go over Chargaff's rules, which is pretty straightforward, and DNA replication, which is also very straightforward. So bear with me, we're almost done. Alright, so, right here. What are Chargaff's rules, right? So I'm going to go ahead and give you a problem, and I want you to work on it. So, I'm going to tell you that you have 10% C, right? And I want you to tell me what percentage of G you would have. What percentage of T? Okay, so I'm going to go ahead and give you 2 minutes to work on this. This is a pretty good question. I would expect to see something like this on your final exam. Okay, cool. Alright. Yes, in the back, left. Yep, that's it. So, how would you get that, right?
That's what I did.
So, how much T can you expect to have in your DNA? So we know that in Chargaff's rules, basically that means that the amount of C is going to match the amount of G, and then the amount of A is going to match the amount of T, right? So they're in equal amounts. So what you can expect is if you have 10% C, you're going to have 10% G, right? And if you have 10% of each out of 100%, that means that you're going to have 80% left over for A and T. So what does that mean, right? Now if you have 80% left for A and T, you split that in half, and now you have 40% for A, matching up with 40% for T. Okay, cool. Alright, so we went over that. Let's go ahead and go over our last topic for today, which is going to be conservative replication and DNA replication. Okay, cool. Alright, so let's go ahead and talk about this, right? So we have our DNA strand. So this right here is DNA. And step 1 is going to be unwinding the DNA, right? So step 1 right here is performed by what enzyme? How do we unwind the DNA? What unwinds it? Yes? No? What is unwinding? What's breaking the hydrogen bonds? No? Helicase, that's right. So, unwind DNA via helicase. So, you have helicase, and we're just going to give this a shape, right? We're just going to say that it's a triangle. Helicase is going to come in. And it's going to unwind our DNA and separate it into separate strands. And what is it breaking in order to unwind the DNA? It's breaking what kind of bond? Yeah? It's breaking the hydrogen bonds, that's right. Breaks hydrogen bonds. Holding nucleotides. Holding nucleotides together. Okay, cool. 2. right here, we have nucleotides being separated into separate strands, right? So what is going to happen next? Something needs to come in and lay down the foundation. That thing is primase, right? So primase adds an RNA matching section to DNA, and this allows for elongation, for duplication. Okay, so primase is going to come along, right? Step 1, step 2. We're going to go ahead and, for primase, right, we're going to go ahead and put a, we'll call it a square, right? So primase is a square. Primase is going to come in, and it's going to put these primers on.
That's what's going on there. Now our next step is what? We need to actually build upon the primase. So what does that? That's right. So right here, 3, DNA polymerase. Adds and elongates on the existing strand. Okay, 4. Now, in order to actually like export your DNA, you need to get rid of all the mess ups, you need to get rid of all the little cuts and mistakes that might have happened. So what do we do to seal it all back together? There's something that we use. What is that thing? Right, so ligase helps seal DNA back together. Okay, so step 1, split the DNA. Step 2, add the primers. Step 3, we're going to build our complementary strand, right? Let me get my red pen. Let's see. Here it is. So we're going to build this complementary strand. Okay, step 4 is going to be, that's 3. Step 4 is going to be sealing everything back together. So step 4, this thing that looks like a glue bottle, right? Okay, so that is what's going on there. Alright, so what is semiservative replication? This is our last thing. Semiservative replication basically means that you're using your original strand and you're breaking it into 2 separate strands and then you're adding on to the existing strand. Semi-conservative means that you're using one piece of the old strand, and you're adding on to that old strand. Now, what is not semiservative replication? So basically it's not this, right? So I'm going to put a giant, like, X on this, and I'm going to write inside. What it's not, and this is really tricky, this is what they were thinking about at the beginning when they were thinking about DNA replication. They were like, okay, we know that DNA has to replicate, we know that it happens. Is it doing semiservative replication, which is like this? Or is it doing something where it's taking the original strand and keeping it intact, but making this right here? So go ahead and look at this picture. You can see right here that you're using the original strand and you're keeping the original strand together, and then from the original strand you're making a duplicate strand that's like this, right? So it's not this. What I'm telling you is that you're taking the original strand, you're breaking it into 2 pieces, and you're adding on to those 2 pieces. So that's where DNA replication comes into play. The top one that does not happen, this one is conservative, and you don't need to know the name, I'm just telling you. And the one that happens, right, the phenomena right here where you build upon the new, or the old, build upon existing strand. This right here is semi-conservative. Replication. Ah, so I lied. There's one more thing that we didn't do. I know. Okay, this is the last thing I
Hate to
promise.
see it.
And we can do it really
Hate
quick.
seeing that.
What are the 3 differences
Really hate
between
it.
DNA and RNA?
I know what that is.
The first one right here,
I might go back.
3 differences between RNA and DNA. I'll go ahead and write a little chart right here, right? So we're going to look at this. DNA uses deoxyribose, which is the sugar, right? And RNA is only going to use ribose. Right here, DNA is double-stranded. RNA is going to be single-stranded. DNA is also going to have A, T, C, and G, right? So we know that. RNA is going to have A, U, C, and G. Okay, cool. All right, so that's it. Thank you so much for respecting this recording. I hope it's helpful for you over the weekend and when you're preparing for your exams. So yeah, good job.